So in the previous lesson, we say that action potential is always positive 30 millivolts, right? It's always positive 30 millivolts. Now, in this experiment, you can see that there are three curves in the graph, okay, for A, B, and C, three scenarios. So in this experiment, the axon of the squid is being placed in salt solution of different concentration, okay? So can you tell me between A, B, and C, among A, B, and C, which axon is being placed in the highest concentration of salt solution? So most of you say A, right? So from here, you can actually roughly guess why is it for us the action potential is always positive 40 millivolt. That means it's governed by the sodium ion concentration outside the cell, isn't it? Inside the tissue fluid. So it's governed by sodium ion concentration. So you can see that in this experiment, when they use higher concentration of salt, then the membrane action potential is higher. When they use a lower concentration of salt solution surrounding the axon, then the action potential becomes lower. So that means it tells you that it is a sodium ion concentration in the surrounding of the axon that governs the action potential. And then in us, right, the sodium ion concentration in our tissue fluid is actually controlled at a set point, remember the norm, by homeostasis. Right, tissue fluid is considered as internal environment. And you learn about homeostasis already, that our body will always maintain a constant internal environment, right, at a set point, regardless of external environment. So because of homeostasis, our sodium ion concentration is always constant or within a range, a normal range. So because of that, when the sodium pores open, right, because of this concentration, the rate of diffusion, this one you learn it, the rate of diffusion is actually governed by concentration gradient, and this concentration gradient is always the same because the sodium ion concentration inside tissue is always the same. So it's governed by concentration gradient times surface area divided by distance. So because the concentration of sodium ions in a tissue fluid is always maintained at a set point by homeostasis, so concentration gradient will be always the same. So therefore, the total sodium ion that can diffuse into the axon per unit time, because it's a rate, isn't it? Rate means per unit time, right? Will be the same. So therefore, the action potential is always the same at negative 30 millivolts. Okay, make sure you write this into your notes. This is a possible exam question, okay, to ask you to explain why it's always positive 30 millivolt. I guarantee this question will come out exam, but I do not know when. Okay. Hopefully, it's your A2 exam. Okay. Right. So now we're going to look at the property of action potential. Number one, all or nothing law. Number two, stimulus intensity by frequency code. Number three, speed of conduction. Number four, frequency of action potential. So this graph shows you, the graph on the top shows you the membrane potential of the axon versus time. Versus different experiment. The bottom graph here shows you intensity of stimulus. For example, the loudness of the sound. Okay, the loudness, intensity of the sound. So there are sounds that come to your ear, okay, but because it's the intensity of the sound is below the threshold, so your stimulus also got threshold intensity on. So when the sound is below a certain loudness, so we say below a certain threshold intensity, we will not be able to hear it, because there's no action potential generated along the neuron to send the message to the brain. So no action potential, even though there's stimulus. Then above the threshold stimulus intensity, see the sound becomes louder and louder above the threshold, which is probably this line here. Then every time the stimulus comes in, the action potential can be generated. So this property is called all or nothing law. either you get the entire action potential positive 30 millivolt or you get nothing. And you can only get the positive 30 millivolt when the intensity of the stimulus is above the threshold intensity for stimulus. Okay, so this threshold for stimulus, not the threshold for membrane potential, it's different. Okay, so this all or nothing law. So here you can see that this width of arrow shows you different intensity of stimulus. This intensity is definitely below the threshold, so there's no action potential. This is above threshold, therefore there's action potential. And between these two, this is actually a stronger stimulus intensity compared to this one. So how do we tell, how can we tell that this is stronger stimulus? Because action potential is always positive 30 millivolt is by the frequency of action potential. So the stronger is the stimulus, the more action potential will be generated per unit time. So that will tell us the difference in the intensity of the stimulus, okay, by frequency of action potential. So intensity by frequency code means frequency of action potential, okay? So presence of myelin sheath will actually increase the speed of conduction, because whenever there's myelin sheath, right, the action potential can only be present at the node of Ranvier, because by now you know already the membrane potential can be produced because of the movement of sodium and potassium ions across the membrane. So in the region of the axon that's covered by myelin sheath, like here as well as here, no ions can pass through this myelin sheath, either going into or out of axon. So this process cannot happen. So this region of the axon has no membrane potential, it's zero. I cannot say no membrane potential. The membrane potential here is zero, okay? So the only place where there can be movement of potassium and sodium ions are in the node of Ranvier. So therefore, when action potential is generated here, the next part that it can generate action potential is the next node of Ranvier. So therefore, when action potential is produced here, then straight away, at the next instant, the action potential will be produced here. So it jumps from node to node faster than when you have to move from one point to the next point along the axon. It'll be slower. Okay, so without myelin sheath, the action potential has to travel from one point to the adjacent point along the membrane, so it becomes slower. Okay, so presence of myelin sheath will cause action potential to jump from node to node. Okay, now diameter of neuron will also affect the speed of conduction. Okay, so the higher the diameter, like for example, I'm going to draw this is a larger diameter of axon. This is a smaller one. So the larger diameter, you can see that what happens is that there'll be more membrane, isn't it? You need more membrane to wrap around this larger diameter than this one. So when you got more membrane, that means there's higher surface area of cell surface membrane. Okay, so when you got higher surface area of cell surface membrane means more number of voltage gated sodium potassium channel proteins. So which means to say at any one time, there'll be more sodium ions or potassium ions that can diffuse across the pores that are open, either during depolarization or repolarization. So therefore, the membrane can reach action potential faster. It can reach back to resting potential faster. So that means to say that it can send the action potential faster along the neuron. Okay? So that's why when the diameter of neuron is higher, the rate of conduction of action potential also becomes faster. Okay, so this is jumping, action potential jumping from node to node in a myelinated neuron, and action potential that cannot jump from node to node, so you have to pass from one point to adjacent point along the unmyelinated neuron. So speed of conduction for myelinated is 100 meters per second, whereas for unmyelinated is 0.6 meters per second. Okay, it's a huge difference, right? Huge difference. Okay, so jumping from node to node is considered as saltatory conduction. So please learn up this term, saltatory conduction. Okay, So you understand the local circuit when I teach you the formation, a transmission of action potential. Okay. All right. It's time's up already, so I have to stop but I will continue to do my recording. So you can leave, okay, to go for the next class, but I'll continue the recording. Huh? All right. So this table actually shows a compare between myelinated and non-myelinated axon. So myelinated axon action potential jumps from node to node. So this is called saltatory conduction because saltare means to jump. So the local circuit is long. That means whenever the sodium ions move, right, it actually creates a circuit. I will explain this one in the next lesson, okay? So the local circuit is long. It's from one node of radial to the next, okay? So that gives faster conduction. Maximum speed is 100 meters per second. You write this in exam, you get 1 mark if you memorize it, okay? Non-myelinated axon, action potential travels to adjacent membrane only, so the local circuit is very short. So between one point to the next point along the membrane. So therefore, there is slower conduction. Maximum speed is only 0.6 meters per second. Okay, so this is myelination, will increase speed of conduction, same as when you increase the diameter. Please cancel off reduce resistance to flow. When you increase diameter, you increase surface area. So you increase the number of sodium ions and potassium ions that can diffuse across per unit time. So increase the rate of action potential produced, therefore increase the rate of conduction of action potential. Okay. So these two properties will actually affect the speed of conduction of action potential along the axon. Okay. Now this graph shows you the there are two lines here. Okay. So it shows you the diameter of unmyelinated axon and the diameter of myelinated axon and the speed of conduction, so conduction velocity. Okay, so you can see that for both of them, when the diameter increases, the speed of conduction also increases, correct? Okay, and also you notice that for yeah, correct. You see, for unmyelinated neuron, right, the speed of conduction is always lower than no, actually not true. You see, for unmyelinated neuron, when the diameter is less than 400 for unmyelinated neuron, the speed of conduction is actually faster than myelinated neuron. So that means to say there's an advantage to have unmyelinated neuron when the unmyelinated neuron is very small. Then it's able to conduct the action potential faster when it's very small. But when it's very big, it's better to have got the myelinated neuron because the conduction of action potential is actually faster. Okay, so this graph also you can use to explain when is it that unmyelinated neuron is better than myelinated neuron, and when is it that myelinated neuron is better than unmyelinated neuron in the speed of conduction. Okay. So refractory period, I will teach you refractory period when we talk about in the next lesson, when we talk about the transmission of action potential. So now I'll skip this part here first. Okay, so what you have learned in this lesson is you have learned the formation of action potential, which involve resting potential, depolarization, repolarization, and hyperpolarization. You have not looked at transmission of action potential yet. It's the next lesson. And we have looked at the properties of action potential, which is all or nothing law, stimulus intensity by frequency code, speed of conduction. So this frequency of action potential, I will tie it together with the transmission. So this one will be in the next lesson. So that's all for today.